Me again. Okay, I'm going to go through your revision guide with you. Okay, so if you have your revision guide in front of you, get it out now. I'm on page 13. What I'm going to do is quickly go through each page and explain different things to you. Okay, let's rub this off the board. Genetic testing. Now, genetic testing quite simply means testing the DNA of a fetus, an embryo, a child, or an adult for certain genetic disorders. Okay. Now, it says in embryos, okay, and fetuses. Remember, a fetus is a slightly further developed version of an embryo. There's two main tests that we can do. These are called PGD and amniocentesis. Now, the key difference is that PGD, okay, is tested before the embryo is actually implanted into the woman's uterus. So what happens is this. The woman conceives or has the embryo by IVF. Okay? IVF stands for in vitro fertilization. And quite simply, what happens is the woman's eggs are removed from the uterus, like so, okay, and then they fertilize with the sperm actually outside of the body, in the test tube, so to speak. And these form embryos, like so. Okay? Now, what PGD does is it will remove just one of the cells from an embryo and it will test that cell for signs of a genetic disease. If that cell shows no genetic disease, what will happen is that embryo will then be implanted back into the woman who has the baby as normal. If that embryo is shown to have a genetic disease, what they will do is discard that embryo and test on another one. Okay? So that's PGD. So in other words, we test these embryos and discarding the ones we don't want before they actually are implanted into the female. Now, amniocentesis is slightly different because at this point, the fetus, excuse the back diagram, the fetus is already growing inside the woman's uterus. Okay? And it's in this amniotic sac. And what they do is they place a needle through like so, and they just take a sample, okay, of the liquid here. Because the liquid here will contain fragments of the baby cells. And then from that, they can test again genetically to see if it's carrying a disease. The difference here, of course, the baby is already grown inside. Okay. To obviously looking at abortion. In this case, we're not technically looking at abortion, are we? Because the child hasn't technically been conceived or grown yet. Okay. So this brings up ethical issues. Okay, ethical issues basically means, is it right to do these types of things? And different people are going to have different views. Many people, in particular many religions, have views against testing, certainly for testing the fetus, okay, and aborting, because obviously that is destroying the life. But also about testing embryos, because of course all these embryos here are going to be discarded. Okay? And lots of people think that's particularly bad. The other thing, of course, is if you test a child and you find it positive it has a genetic disease, doesn't that mean then you have to test all the other children you may have because they might be carrying the disease? And is it right then to put those children through the same procedure? The other times we test is we can test adults and children. We can take a cell sample and again we can test for genetic diseases. Now remember, what about Huntington's? Remember we had a discussion whether you would actually want to know the results of your test or not. And again, there are ethical considerations, aren't there? Because if you went for a test, and then you've had brothers and sisters, what do? Are you then forcing them, sorry, caretaker's family, hi guys, are you forcing them people then to have a test as well? Okay, so there's ethical issues there. Also, you have to consider your employer, okay, and any health insurance that you may have. Because, of course, if you found to have a genetic disease, how is that going to affect your job and your employability? Okay? The other people, the health insurance, remember, you pay them money and then they insure you against medical treatment. Now, obviously, if you've been tested for a genetic disease, they're going to use this information and they're going to use it against you because they're going to charge more for health insurance because you have a genetic disease. Okay? So these are some of the issues that you have to think about. Alright, over the next page, looking at clones. Now a clone, very, very quickly, it's quite straightforward. All it is the offspring of a single parent. Okay? They're identical in every single way, and it's an offspring of a single parent. And that's it. Stem cells. Stem cells are very, very, very important types of cells. Because what a stem cell can divide to form any type of cell in the body. Okay? Now the most powerful of these are called embryonic stem cells. Okay? An embryonic stem cell can become any other type of cell. As you get older, stem cells become what we call specialized stem cells. Now the specialized stem cells are not so powerful because they can only become a small group of different types of cells. Okay? So for instance, specialized stem cells in the bone marrow can become white blood cells, red blood cells, platelets, etc., etc. But they couldn't become nerve cells. Yeah? Whereas an embryonic stem cell can be used to become anything at all. Now, it is hoped that these stem cells can be used to treat illnesses. Okay, now if you look at the bottom of your book, that's statement 1 and 2 will give you some of the things that stem cells can be used for. In the examinations, you need to know exactly. Okay, and that. Guys, I'm going to have to go home, I'm afraid. So I'm just going to post these videos up, and B2 and B3, you're going to have to look at yourself. Can you do me a favour and in particular look at species and sustainability? Any problems, just send me an email or contact me via the D2D and I'll get back in touch with you. Okay, have a good Christmas and I'll see you again.